All right. Hi, guys. So today we are going to talk a little bit about the software cell chat. So after you've already processed your single cell RNA sequencing data, you've already labeled your cell types, and you don't really know how to analyze this data, cell chat might be a great option for you depending on your goals. So cell chat looks for different cell-cell interactions within single-cell RNA sequencing data. So why is this important? Well, these different cell-cell interactions that we can identify using software such as cell chat can actually give us insight into how these cell-cell interactions coordinate cellular function. So an example of this biological mechanism is shown on the screen here. We have a sending cell and a target cell. The sending cell will secrete a ligand, which will be associated to its associated receptor pair. And once these two ligand and receptor molecules interact with each other, it will elicit a certain type of response. And so cell chat actually leverages single cell RNA sequencing data to evaluate cellular function by looking for these different cell-cell interactions. And so again, the input is single cell RNA sequencing data. So data where we have isolated and sequenced the individual cells. We'll get an expression profile for each individual cell type. And then we can actually compare the profiles of different cell groups. And so how cell chat works is by using a network-based analysis. And so before we dive into how this works, I wanted to talk a little bit more broadly on what a network is and how it works. And so a network is a connected set of nodes and edges. So nodes are these dots in all of these figures, and the edges are the interactions between the different nodes. So on the left-hand part of the screen, on the upper half, we have a network. And in this paper, the authors were using this network to look at the hierarchy in faculty, the whole faculty hiring process at Ivy League institutions versus just general public institutions. Down here, we have a network from a paper that was looking at how COVID-19 spreads. So they used a network-based approach to assess how COVID-19 spreads through a community. And then up here, we actually have an output from cell chat. So the different dots in this circle are the different cell types that we have pre-labeled before running cell chat. And then you can see the lines in between these cell types are actually the interactions between these cell types. So a thicker band indicates that these two cell types are more likely to be interacting with each other than a thinner band between two cell types. All right, so how does cell chat actually work? So the first thing that cell chat does is it cross-references a ligand receptor interaction database. So the creators of cell chat have actually created a database called cell chat database that is a manually curated and validated signaling molecule interaction database. So essentially, it's just a framework from which ligand and receptors interact with each other. The next thing that cell chat does is it infers and visualizes these different intercellular communications. So it identifies differentially overexpressed ligand receptor pairs, and it builds this network where the nodes are the cell types and the weighted edges are the cell-cell communication probabilities. And then the last thing that cell chat does is it performs quantitative analysis. So it determines the major signaling targets, sources, mediators, and influencers. So how it does this is it calculates the in degree and out degree for these different nodes. It will calculate degree centrality and betweenness centrality across these different networks to get a good idea of which cell types are interacting with each other. So these are the different outputs that cell chat outputs. You can get a hierarchy plot, a circle plot, which we'll be doing today, a bubble plot, So we're not going to make all of these different plots, but depending on what your goals are when you do single cell RNA sequencing data, the cell chat vignette on the GitHub is a great resource and has lots and lots of information on how to make these plots. So now I'm going to briefly talk about how to run cell chat, and then we'll do a little coding demo. So the first thing that you want to do is load in your processed Seurat object. You can load in an object that has not been labeled, and you can let cell chat label your cell types for you. However, I would not recommend that because as we have learned from our cell labeling day, there are a million ways to do this, and I think it's best to just do it yourself so you really understand what you have done. All right, the second thing that you can do when running cell chat is to convert this cell chat object or this Seurat object into a cell chat object. You then will calculate these different cell-cell communication probabilities. So essentially what this is doing is looking at the expression of the ligands and receptors within each respective cell type that you've pre-labeled your data with. And from looking at these different expression profiles, CellChat will use its networks-based analysis to tell you which cell types are most likely interacting within your data. And then you can actually visualize your results. So here's an example output for the CCL signaling pathway network. This D21 right here is just indicating that this is a disomy 21 individual. The dots around the outside of this plot are the different cell types, and then again, these different connections between the cell types are the different edges. And you can also get heat maps from CellChat, which is really, really helpful. This is one of my favorite plots that you can make with CellChat. It's really, really informative because it can tell you within this signaling pathway which cell types are most likely a sender, a receiver, a mediator, or an influencer. All right, so now we're going to perform the coding demo and actually run cell chat ourselves. So the first thing that we want to do is load in the required libraries. One thing I want to note is that I am working on the AWS. If you guys have successfully installed cell chat and Seurat, you're going to be running this on your local computer and downloading the respective data from the Zenodo. Another thing I want to point out is that if you are on the AWS, make sure that you are using our version 4.4.0. All right, so let's load in our required libraries for cell chat. All right, now we are going to be loading in this pre-made Seurat object. In the real world, you guys will be making your own Seurat object, performing your own cell labeling, but for today, to keep everybody on the same page, we're all going to be using the same objects. So we're going to perform this using this load command, and that will take a second to load into our environment up here. And we're going to start with the trisomic data just because it's a little bit smaller and it will run a little faster. But if you finish the worksheet early, you're going to run the disomic R data, which is a Seurat object located in the same folder. All right, so the second thing we want to do is actually create our cell chat object. So we're going to do that by running this create cell chat command. And now we can see our cell chat object is shown in this data in our environment. Now we're going to initialize the cell chat database. Another thing to note is that you can run cell chat on non-human data, such as mouse, but we're not doing that today because this data is human data. So this line, this show database category, is just going to show you a little bit about what makes up this database. So you can see the database is made up of secreted signaling, ECM receptors, cell-cell contact, and non-protein signaling pathways. There's more information. You can actually look at the vignette to even get more information about what makes up this database. And then we're actually going to take a look at what this database structure looks like right here. And you can see we have the interaction name, the pathway name, the ligand receptor, a bunch of great information in this database. So the next thing that we're going to do is add the cell chat database to the cell chat object. Today, we are only going to be looking at secreted signaling pathways. However, when you run this yourself, you can run it on the whole database or your preference for ever what you're trying to do. All right, so now we are going to identify highly variable ligand receptor pairs. In this step, cell chat identifies these highly variable pairs, and it looks for differentially expressed ligand receptor pairs across cell types. So this step requires a decent amount of memory on the computer or the cluster that you're working on. So I recommend running this future plan command, which essentially helps the software run in parallel. And now we are going to identify the actual overexpressed genes and overexpressed interactions. Just to note that this will take a couple minutes. I think on the AWS it takes anywhere from 3 to 5 minutes, but just hang tight. All right, so our identify overexpressed genes and overexpressed interactions command ran, and we can see that the number of highly variable ligand receptor pairs used for the signaling inference is 2,653. So you're going to fill that in here on your worksheet. And then now, if you were running this not in the real world, this step takes a little bit longer. I think it takes up to an hour sometimes on local computers. And we cannot run it on the AWS cluster because this cluster is just too small. However, when you are saving the script for future reference, what you would want to run is this compute communication probability. So essentially what this does is it looks at all the differential ligand receptor pairs, and then it computes this communication probability. So the likelihood that two cell types are interacting through a specific pathway based on the expression of the ligand receptor pairs while cross-referencing that database that we have added to our cell chat object. However, we're not doing that today, so I'm actually going to just comment that out. So now, when you guys are working through this in class today, after you have completed the end of cell chat part 1, you're going to move on to the cell chat part 2 worksheet, and we're going to clear our environment. And so because that step takes so long, I've actually already ran it on my computer, saved it, and so you guys can just load in the already processed data without having to wait an hour on the local computers and also without burning out the AWS cluster. All right, so again, we're going to load in our respective libraries. And then we're going to load in this trisomy 21 cell chat object. And you can see it up in this left corner. All right, and so the first thing that we want to do is filter out any cell-cell communications where the probability of the two cell types interacting was quite low. So we don't really want anything, these are like false positives. We don't want any false positives to be represented in our network plots that we're about to look at. Now that we have removed some of these cells, we are going to recalculate this communication probability pathway. However, this step will take a lot less time because we've already ran it once. And so now it's just allocating the fact that we have removed some cells from our data. All right, now we are going to count the number of edges on a per network basis. So this is the aggregate net command. And now we're going to set the group side. And now we're going to actually plot this circle plot. All right, so this looks pretty crazy. But you can actually click this zoom button and you will be able to get a better picture of all the interactions. However, one thing to note, we can actually see it by making this bigger, I bet. One thing to note though is because there's so many different interactions occurring within this single cell RNA-seq data cell chat object, it's really hard to get an idea of what's going on. And so one thing that we can do is look at a specific interaction profile for a given pathway. But let's say you're analyzing your single cell RNA sequencing data and you're not sure which pathway to look at, which has happened to me before. So what we can do is actually generate one of those heat maps that I showed in the slideshow. And from these heat maps, we can select different pathways that would be interesting to look at. So let's run this. So we're going to generate a heat map for sending cells and receiving cells. And a way to show both of these heat maps at the same time is to run this ht1 plus ht2 command. So this ht1 is these are the outgoing, so the senders, and then this ht2 is our receiver cell types. And now we've generated this beautiful heat map that we can save onto our computer and get an idea of which pathways that we should be looking at. And right here, you can see all the pathways are on the left here. It looks crazy because we had to condense the image to just our little tiny plot portion of this RStudio environment. However, you could download this image and actually look at the different cell types by blowing it up. All right, but for today, since we are working with Down syndrome data, I guess we might have to leave this like this for now. There we go. All right. But since we know that we're working with Down syndrome data, we actually have an idea of some pathways that we would be interested in. And some of you who work with Down syndrome might know that interferon-related pathways are really of interest to Down syndrome researchers, since many of the interferon proteins are located on chromosome 21. So I have a list of pathways that you guys can analyze using cell chat. So today we are just going to look at the CCL pathway. However, you can check out all of these pathways when you're running this yourself. So this sets the pathway as CCL. And we're saying, okay, we are going to plot a single plot. This is this plotting layout command. And now we can actually generate one of these circle plots. And it's much, much, much easier to look at compared to that network that was of the entire single cell RNA sequencing data. This is just for the CCL pathway. You can actually do the same thing for the CCL pathway. However, just look at it in the heat map format. And that's this net visual heat map command, which is really helpful looking at the different communication probabilities between cell types through this CCL signaling pathway. And you can actually look specifically at the contribution of each ligand receptor to the overall signaling pathway. So you can do this by this net analysis contribution. Look at the contribution of each ligand receptor pair. And one really cool thing that you can actually do is select specific senders and receivers to visualize. So here we're just looking at this natural killer cell as the senders, and then these CMP, DC1, and DC2 as the receivers. And you can actually see that in this x-axis. You can mess around with this. You can limit which pathways are shown. So here we're limiting the pathways to the only pathways to be shown are CCL, CXCL, and TNF. And you can see that TNF is the only one that pops up here. And then my recommendation from here is to just look at the vignette and really get an idea of the different options of visualizations that you can make for your single cell RNA sequencing data. And then another idea is to take the D21 object, plot the exact same plots that you made for a single pathway, such as CCL, and look at the differences and see what you find. I think cell chat is a great way to analyze single cell RNA sequencing data when you're looking for the presence of different cell-cell interactions.